Niab is the lead organisation. That means ultimately we're responsible for ensuring that the programme is delivered. And we're here on the campus with the East Malling Trust. And the Trust is a really important supporter of what we do now and will be really important in our plans going forward. So we've engaged with over 700 businesses. We've done over 100 projects. And many of you, I suspect, may be involved in those projects. And we're really pleased with how those projects have gone. And we've managed to create something, I think, in the last 5 years that offers you both the opportunity for a technical discussion, but in other areas of work, it's also around collaboration, and it's very much around mentoring and business support. So it's quite a diverse sector that we program that we lead. One of the challenges I think we all face is the brilliant new ideas we're going to hear about today. How do we actually get those into our everyday practice? How do we actually make those become part of our normal routine of when we're working? And for me, that's been a significant challenge. I worked in a university for 7 years, and I've also worked in industry for the rest of my time, probably for about another 28 years or something like that. So I've seen very much both sides of what I call this wall and some of the challenges in that wall. And you're lucky today that you're going to hear some really good technical presentations that are going to help you get an understanding of what technology is coming down the road. And we want to make sure you continue to hear that opportunity. So, but also potentially you get the opportunity to get involved in the design of any new equipment or ideas or systems. You get involved in the processes. You need to understand the implications of regulation, and you need to be able to maybe trial those products on your farm or at your place of work, or indeed trial some of those ideas here at East Malling. So my point really is there's a lot of work that connects the ideas we're going to hear today to what you see in the field. And our ambition is really to do a number of things. It's to de-risk the investment in innovation. It's to enable the scaling of those ideas to happen more quickly, so you're able to do more quickly, and it's able to bring forward some of that technology. Now, I think what I'm also aware of, having worked in the sector, is that this is an interesting time for the sector, moving significantly from a chemical-based, carbon-based how are other people using it becomes even more important in the next 4 or 5 years. And again, that's why I think an organisation like Growing Kent Medway, working with Niab and the East Malling Trust, can offer you something that can really help you in that transition to regenerative farming, for example, and using less carbon and chemical-based solutions. So understanding how this works, I think, is really important, and having the option to go to workshops, to join sandpits, etc., is of value. The other thing that we want to do at Growing Kent Medway going forward, and it's something I think we're increasingly aware of, is that the importance, and let's look right on the right-hand side of this chart, of consumers. Consumers do drive change, whether we like that or not. It's sometimes a very positive change and sometimes it's a negative change. But effectively, when you're talking about something like soft fruit, which is highly visible, has high qualities of nutrition, is potentially easily accessible, it's in a format that's easy to adjust, it has a really important role to play in reducing the impact of poor food on our health system. So DEFRA talk about something called a good food cycle, which is all about producing that food well, being able to put it through a process well, not to lose the nutrient value, retailing it in a way that becomes attractive and affordable, and encouraging people to consume that food. I was at a REATing conference where there was quite an important presentation by Professor Chris Whitty, and he said we need to eat 30% more fresh fruit and vegetables, and we need to eat 50% more fiber. Now, he used the expression, and this was a conference largely for food manufacturers and retailers, you as a group need to lean into this challenge. You need to ensure that you are changing and the way that you're changing is visible and that the health service can see this change. There have been some startling figures of the impact of poor food on the health system. I've seen figures ranging from something like £11 billion a year, £40 billion a year plus of the impact of the diet, our diet and our food consumption on that. And in Kent, we have a real diversity of wealth and deprivation. In East Kent, there are real challenges around people's diet, and this is not uncommon for coastal communities. And being an island, we are particularly subject to this. The other interesting comment I heard, and apparently this was a view from Treasury a long time ago, was that we don't need to worry about food resilience. We're a wealthy, top 10 global economy. If we don't have the food in the UK locally, we'll just buy it internationally. In other words, we can afford to buy it. We don't have to worry about producing that food in the UK. It's not a problem, we'll just import it. And that was a view from Treasury. I think, well, I know that view has changed. Talking to retailers, for example, they are now talking about resilience of their food supply chain, particularly with the view on tariffs, climate change. They want to be able to have food on those shelves. Tesco still has the ambition to increase its market share to 30% of the UK grocery market. To do that, it needs to have the product on the shelves. And what I think is happening increasingly with the retailers, they need to understand the vulnerability of their supply chain. They need to understand how some of the huge increases in productivity that you've seen in the soft fruit sector in the last 20 years, how can that continue to enable them to increase the resilience of their supply chain? So the reason for talking about a food system, and again, DEFRA talk about a food system, is everyone in this room is an inherent part of the food system. The consumers are at the far end. And what we have to do as Growing Kent and Medway and as policymakers is really encourage people to transform that food system, to increase its resilience and increase the content of UK supplied fruit and vegetables. So I just want to talk about Local Innovation Partnership Fund. So this is interesting for a number of reasons. It's a £500 million fund. So you will talk a lot today about research and you'll talk about innovation. Now, traditionally, the UK government has funded that across the whole of the UK. It's run open competitions. What we are seeing a change to is what's called place-based innovation with national impact. So what the UK government is saying is, for example, we wouldn't accept a shipbuilding company to start up in Kent. If you're going to build ships, you're probably going to do it in Belfast or you're going to do it in Glasgow. And similarly, maybe if you're going to start some kind of innovation around dairy, you're more likely to do that in Southwest Scotland, the Lake District or Devon than you are Kent. So there's a focus really on where they put the money and how they encourage some of the things I've just talked about innovation. This is now becoming part of the strategy of devolved administrations. So mayoral authorities will have funds to support their local region and their local industry. We're already seeing that happen. Our colleagues up at Niab in Cambridgeshire are already under a devolved authority with Cambridge and Peterborough, and life sciences, needless to say, in Cambridge is one of the key sectors. I'm pleased to say that in partnership with Kent County Council and Medway, food and agri is one of the key sectors for this particular region. And for us, that particularly means horticulture, soft fruit, top fruit. So we have, over the last year or so, made a really important opportunity for investment in innovation in this region. And so we're applying currently for a £20 million fund over 5 years. To do that, we need £20 million of match funding. So this would be a total fund of £40 million. Now, there's an opportunity to do a lot with that 40 million, and we look forward to doing that. In order to capture the imagination of politicians, capture the imagination of universities and colleges, and capture the imagination of you in the room as well, we have come up with what we call 3 platforms that we want to base the future opportunity for horticulture and wider sector in the UK. And we've learned from the food system and our work. So platform 1 is around smart and resilient horticulture. How can we ensure the resilience of the supply chain? We continue to increase crop yields. We reduce the risk of disease and pests. We support regenerative and precision farming, and we strengthen supply chains. We are also very keen to understand how we generate a bioeconomy through things such as biochar, producing biochar at scale, biorefining, looking at the challenges you all have on farms and places where you work for energy, water, people. So how can we match that into your ambition for changing the way that you deal with a lot of challenges? And the final platform is around functional food. So that is really around how do we design food that is highly nutritious? How are we bioengineering products that may be a waste product to you, for example, because it may be damaged or bruised, into something that has a high nutritional content. It's a very ambitious programme, but it has the support of the county councils, all of the universities in Kent, and is currently being led by Growing Kent and Medway. And we will collaborate with the East Malling Trust here to make sure that this particular site becomes the home of the smart and resilient horticulture platform. We are engaging with other institutions across the UK. So the last thing I'm just going to talk about is go back to this concept of place-based innovation with national impact. So this is all talking about doing something, enabling that scaling up, de-risking that innovation, bringing that idea to fruition earlier, that can be done here in Kent and Medway potentially, but then is shared with the rest of you to give you a national impact. If we don't demonstrate that national impact, we won't get the funding. It's that simple. So it's really important that events like today and the number of people we've got online give us the opportunity, and I mean all of us in the room, to share our work and innovation with others. And this is a real opportunity for the UK. You know, we've seen this kind of thing happen in Food Valley in the Netherlands, Food Valley in Switzerland. We see it happening over in Canada and the US. And I'm hopefully now that we can really develop something in the UK that gives you real national impact and helps all of you in terms of that transition to a renewable, resilient, regenerative farming and growing perspective, and helps us increase the amount of consumption of soft fruit at an affordable, accessible price. Thank you very much, and I'll leave it there. Thank you.
Thank you very much indeed, Simon. First of all, can I just say to those of you who are online, please accept our sincere apologies that you were muted in my introductory remarks, many of which were directed at you. Can I just also say that if anybody's interested in looking at the full programme and you're online, I will be going through the programme as we proceed through the agenda today. But if you do want to have a look at the whole thing, you can go onto the Niab website, niab.com, click on events, and you will get the whole programme for today there. Thank you. Any questions for Simon from either the floor or online? Nothing online. Simon, just one thing from me.
Yes.
I don't think you mentioned this. Apologies when I dashed out. But we did speak recently about life expectancy in Kent.
We did, yes.
You want to just share that fact? Because I found this a staggering fact.
So within Kent, this is very detailed life expectancy down to individual postcodes. The difference between a male life expectancy between someone who grows up in the Maidstone and Sevenoaks area and someone who grows up in Sheppey is between 16 and 17 years for male and female. That's within a distance of about 30 miles. So if you grow up in Sheerness, your average life expectancy can be up to 17 years shorter than if you grow up in Sevenoaks. And so, you know, one of the reasons for that is poor diet, poor living environment, and access to healthy, affordable food. So there is a challenge around this nutrition. As Chris Whitty said, the industry needs to lean into that challenge. Thank you, Scott. I have to, apologies, I have to disappear now, but I'll be back at lunchtime to talk to any of you. Please enjoy the rest of the day, and thank you again for coming. Thank you.
Thank you very much, Simon. I'm just going to ask the technical issue, Mark. The image seems to have frozen. That's okay. We're back up and running. Okay, so our first speaker of the main first session is Francis Wamonje. We move on to crop protection now, pest management and soft fruit storage. Francis is known to many of you. He is an entomologist working in Michelle Fountain's pest and pathogen ecology team here at Niab's East Malling site. Large raspberry aphid has been a major problem for several years now. and fewer and fewer insecticides and aphicides to control those pests. And Growing Kent Medway, which you've just heard about, has been funding a number of projects in the last 2 or 3 years, one of which has been led by Francis, and he's going to tell us a bit more about what he's been finding, looking at various biological control options for large raspberry aphid. Francis.
Yeah, thank you, Scott, and thank you everyone who's come to attend and to listen to this talk. Yeah, this is one of the projects which, as Scott has mentioned, was funded by Growing Kent Medway. We had a partnership from industry, Asplings, and Charlton's, who availed their grower sites for us to use in experiments, and BioBest, who are major producers of biocontrol. And together with us, we came and formed this consortium to try and tackle the large raspberry aphid. This project really came from conversations that we had here, and growers saying that they were having challenges with the large raspberry aphid. This wasn't a problem before, but like many other pests that we are observing now, with the deregistration of actives, they are beginning to re-emerge. And there's also the shift towards IPM and much greener technologies. And so we need to stay at the forefront of this. So that's the impact of the large raspberry aphid. But just to put it in context, the UK produces quite a bit of raspberries, but only 30% of what is consumed in this country. So there is a huge scope in terms of production. And this aphid will cost between 10 and 20% of losses. And you'll all agree with me that losing 14 to 28 million a year is quite a significant chunk of money to be losing to aphids. So the challenges are, as I've mentioned, the progressive withdrawal of pesticides has left very limited options for growers. Previously, and I think quite a bit of work which was done by researchers here, there was genetic control for aphids, which was introgressed into many of the varieties. But over time, as growers began to cycle their crop much more seasonally, those resources have since been lost. So control is really premised on biocontrol products. We've got parasitoids and generalist natural enemies, usually green lacewings and brown lacewings. And these are usually deployed manually where they spot outbreaks, which you'll all agree with me is quite labor intensive. So what we wanted to do for this project is to develop an integrated biological control program for raspberry that provides control across the entire season. And we wanted to test a few components. We hypothesized that, yes, there are parasite mixes in the market, but nobody had really investigated whether the mixes in there are fit for purpose in terms of the species composition and even when they're deployed. And so we worked with BioPest in that space. We also wanted to test whether we could deploy the green lacewing eggs by spraying them as a labor-saving measure. And the last thing that we wanted to know is how early in the season can we deploy the brown lacewing, which is a predator. Now, these tend to be, tend to operate at low temperatures, but nobody really had pinned down what temperature they operate at. And we hoped at the end to integrate this into an integrated biological control program that would give us season-long protection against the large raspberry aphid. So just to get into the data, we did look at the parasitoids and how they sort of changed across the entire season. The good news is, if you look at it from the graphs a bit, is that all the parasitoids that you use in your mixes do exist out there in the field when we observe them. What changes is when they do that. So early in the season, you'll find the Aphidius avi and the ones which you normally deploy are dominant. But towards the end of the season, there is a change. And you find that there are other species which are typically not within the parasitoid mixes in there. And the good news is that we've passed on this information to BioBest. And now it's really up to them to look at what are the best candidates that they can begin to work with, either to have single aphid species mixes for certain times of the season, or to integrate them into the mixes and maybe communicate with agronomists on how they're going to deploy this. We also did quite a bit of experiments with the brown lacewing to look at when they begin to predate. We worked on a temperature gradient from 8 degrees to 12 degrees, which would simulate the early springtime. And we conducted our assessments over 24, 48, and 120 hours, just to look when do they begin to really predate. And when they do, are there significant differences and at what temperature? So we've got 5 replicate arenas for all this and an equal number of control groups because we do want to see the die-off happening just because of nature. We wanted to really quantify this in terms of what we really observed. And this box plots, what they really show is that there's really an effect of both temperature and time. So with increasing temperature, there's more predation, and with increasing time. But when we crunch the data, we then find that at 12 degrees centigrade is when you begin to really see the difference in the predation. So at least from our math, nature is stranger than fiction, isn't it? We do know that at 12 degrees centigrade, we do find significant changes in the predation. We then got the opportunity to test this out in the field, and we had an unreplicated experiment that we did on the site. This site had got a lot of infestation. So if you look at the picture of the canes, they're those small, I don't know if there's a laser pointer here. No, there's none. Black spots. All of these are just, eggs that have overwintered. So it was a site which was really quite, infested, and we wanted to see if we deployed this from springtime, when would we get to see any differences? And it was a bit of a time course experiment. And by the 2nd and the 3rd week of April, we could then see where we had deployed the brown lacewing. There was a dip in the number of aphid numbers that was quite significant. But this experiment is not replicated. I will be coming back to you again, hopefully funding allowing to enable us to have a replicated experiments, maybe with different program. Then these are pictures of Adam about to go to Mars, or rather dressed up and doing a spring experiment. And this was for the green lacewing deployment. So we started here. We wanted to see, when we tried out different types of sprayers, and we wanted to see how many eggs do you recover back when we spray that. It's quite a manual process. And once we were able to both find the sprayer which we wanted, which was the gravity one, and the different stickies, we then went to Steve Greenaway's farm to try it out. And by that time, we had got 2 candidates, which we've just coded as TX1 and TX2, and they're the best ones, and water as a control. We did 20 replicates of this experiment. And the things that we were looking for were, 1, how effectively do the eggs adhere onto the leaves? And 2, do they hatch? Because if they don't adhere and don't hatch, then there's really no use for the work. And so for TX1, you could see it's effective, but the eggs tended to get to the bottom of the container. Adam sprayed them, we could get them on the leaves. But what we noted with this particular product is that the eggs tended to fall off as soon as the liquid had evaporated. So it wasn't quite so good. But for this one, you could see the eggs were really well, suspended in there. They stuck well, and there wasn't much residue left at the bottom of the sprayer. So that was really nice. And then when we looked at the hatch rate for all of them, it was about 40%, which compared to the industry. So BioBest have done these experiments on lettuce, and they typically get around 40 to 44% hatch rate. And for all of us, we were getting around 41% hatch rate. So we are within what the industry has been able to find out. But the take-home message of this is that the key thing is the adherence. The eggs have to adhere, onto the leaves. So looking at all the 3, we now had, achieved at least the testing of the different biocontrol components. So in 2024, we did a limited deployment. By this time, we didn't have any data for the brown lacewing. So this was done with the green lacewing and the parasitoids across, 2 sites. The reason why I'm showing this slide, which shows that we couldn't find any differences, is because this was an unusual year. There were very low aphid numbers, and I'm showing it because I need to come back to you guys again to partner with us to have an integrated biological control over at least 1 season or 2 to really test out all the components. We've been able to nail down the 3 components, how to optimally deploy them, when to deploy them, and what mixes we require. What we need is simply an opportunity to really extensively, test it out in the field. So we did this over 1 year. We weren't able to desegregate because in this year, in 2024, the aphid numbers were low. Last year, they were very high. But that's nature. That's how things at times occur in the field. So I look forward to coming back to you guys and requesting if we can work with you to do an integrated biology control deployment. So just in summary, what have we achieved out of this, the funding that we got from Kent Medway that we were really grateful for? One, we now do know that there are parasitoid species that probably we need to get into the parasitoid mixes, or at least shift when we do deploy so that you've got maximum effect. We've effectively been able to find a product that can be used in the deployment of green lacing eggs by spraying. There is a product available from a competitor, organization, and maybe some of these things do help, with the arguments to get the regulation to have these things out there, and that's something which BioBest is following up on. And in terms of micromas, if you're going to deploy micromas out there, because I know some of you already do that, certainly Steve already does this, then once the temperatures get around 12 degrees, we now do have the data to show that you'll find an effect. And for the integrated biology control program, we do need more data. But there's promise in that. And once we're able to do such a trial, we'll be able to, share that with you about the cost effectiveness and the cost benefit ratio that will come from having such a system. Thank you, and I'll take your questions.
Thank you very much, Francis. Any questions from Francis? Nothing online so far. Anybody in the room? Yes. Can I ask that if you're in the room, if you wait for Michelle, she's coming around with a microphone. And when you get it, if you can put it right up to your lips so that we can all hear.
Okay, thank you very much.
Can you also introduce yourself and the company you work for?
Yeah, it's Adam Tyler from SNA Produce. My question is, you're talking about 12 degrees. Is that a 24-hour average, or was there? And have you got any data on the high-end temperatures where they would stop predating?
We don't have data on the high temperature. That's a 24-hour average, by the way. We do have data loggers. Maybe we need to crunch that data, better, but Adam does have that data, because for those trials, we did have data loggers in there. But certainly at 12, you will see a very dramatic, effect of predation.
Okay, thank you. Question online, just asking about saying surely the MX product would need to get approval and a map number before it can be legally sprayed onto the crop. I think that goes without saying. Yeah.
Yes.
Okay. Thank you, Francis. One more. Let's just time for one more, and then we'll need to move on. Yep.
I'm not sure if I missed it, but you said brown lake were effective use or control from the middle of April. How far long after that? How long before that was the brown lake deployed into crop? How long did it take?
Sorry, I don't quite understand. Oh, how long did we deploy it?
Yeah, how long before you saw the control?
Okay, so we deployed weekly from the 3rd week of March onwards, and every week, and then we assessed on a weekly basis.
Okay, thank you. I think we need to move on. Thank you so much, Francis. Take a rest, because you'll need plenty of it for your next presentation. We're going to stick with the theme of aphids now, and we're actually looking at hoverflies as a way of controlling aphids in soft fruit. Sarah Arnold is sadly unable to be with us today. Sarah is doing some work in Brazil, would you believe? So she's very kindly taken the time to record a presentation for us. So we're going to listen to what Sarah's been doing with hoverflies for aphid control in soft fruit.
I'm Sarah Arnold from the Pest and Pathogen Ecology Department here at Niab. I'm really sorry that I couldn't be with you in person today, but I hope that you'll enjoy hearing a little bit about some of the work that we've been doing with some cool collaborators around improving the deployment of hoverflies in soft fruit to manage aphids. So aphids, we all know about them. We're all probably sick to death of them this year in particular. As we're seeing more and more plant protection products being withdrawn, we have fewer and fewer tools available to us for managing aphids on soft fruit crops. And this means that increasingly we're depending on biocontrols. So we need to be applying those optimally in order to get the best possible performance from them. So things like getting the right time and the right spatial distribution in order to make sure that they're doing what they're supposed to. Today, I will be focusing mostly on the 3 aphid species that affect strawberry most severely, perhaps, black bean aphid, strawberry aphid, and potato aphid. But a lot of the concepts today will be equally applicable to aphid pests of other soft fruits, so for example, the large raspberry aphid. Hoverflies then. There's about 6,000 species globally. There's about 270 in the UK, but not all of these eat aphids. Hoverflies can broadly be divided into two groups: the ones where the larvae are predatory, they're mostly terrestrial, they're moving around on plants and eating aphids. On the other hand, there's the other group where the larvae are essentially rat-tailed maggots, so swimming around in stagnant water, puddles, ditches, things like that. Those ones are not relevant for UK crop biocontrol, but they have other important ecosystem functions that they perform. So amongst the hoverflies, the adults fly well, they're really mobile. Some of them are migratory and can travel literally hundreds of miles. And among the aphid-eating species, the females will seek out colonies of aphids. So they're attracted to the odours associated with aphids feeding on plants. And generally, the bigger the colony, the more exciting it is for them. So this means that your biocontrol is targeted towards where the pest problem is at its worst. So it's ideal. There are currently a few species that are available commercially, so being bred, and you can buy them for biocontrol. But is there a way that we can use them, combine them, and deploy them better? Recapping the hoverfly life cycle. So the gravid female will seek out these aphid colonies, the bigger the better, and lay some eggs on or near them. So here, for example, there's some aphids feeding, and that pale green blob in the middle is a hoverfly egg that's been laid close by to them. After a few days, the egg hatches and the larva comes out, and straight away will start feeding on aphids in that colony. And at their peak, they can eat 50 aphids a day quite easily. So the larva is not very prepossessing, perhaps, doesn't really have a lot of features, doesn't have any legs, but can crawl around on plant material, and they're quite mobile for what they are, and certainly very voracious in terms of their feeding and predation habits. So that is the real focus of what we need for biocontrol. They might not look like much, but they are fantastic. After a couple of weeks or so, the larva will pupate, so it will either stick itself to a bit of plant material or go into the soil, go crispy on the outside as pupa, and start morphing into an adult. After a few more days, the adult emerges, and then both males and females are winged, they fly around, they visit flowers, they mate. They're providing pollination services at this point, so that's a real benefit to a lot of crops as well. And the cycle begins again. There's three main species of hoverfly that are currently available commercially in the UK. So we have Eupiodes corolli and Epicercus falciata. So these are both native UK species. They're about a centimetre long, so kind of medium-sized to slightly larger as hoverflies go. They work well at a range of UK temperatures because in the wild they will be active right through early spring, right through to quite late in the autumn. I mean, on a mild day at the moment, you might still spot the occasional one or two on flowers. The other species that's available is Sphaerophoria rupelli. It's a smaller and more slender species. So that means it tends to be a poorer pollinator because its body is simply smaller and doesn't have a lot of hairs on for carrying pollen. And it tends to perform better at warmer temperatures, so is more suitable for things like glass houses and poly houses as pest control. So personally, I prefer the first two. And I've been working with this company called Alombria. They're really exciting. They've been going for quite a few years now, and they're taking hoverfly technology beyond just the supply of flies. So it's not just the flies that arrive in little boxes that can be deployed into the crop, but they also can provide cameras and monitoring services within the crop. So these tell you about things like how those hoverflies are responding and their activity levels. They're adapting the system for early pest detection on the crop, so looking for when those pests are starting to develop as a problem, and providing wider information about pollination services from all of the pollinators on the crop and other insects that are present on the farm. So there's a whole suite of data that's then provided to the grower through the app and the web portal. And this combined can also feed into deployment of lures. So these are attractive odor-based packets, if you like. And when the system alerts that a pest problem is developing in a particular part of the crop or the facility, then these odor lures can be deployed. These are attractive to the hoverflies, so they'll move towards it. And this means that the hoverflies are moved to where they're needed the most, rather than just having to do lots of untargeted releases and hope for the best. So Alombria produce all of their hoverflies within the UK, so I think that's a bit of a unique feature. They have quite a wide species range that goes beyond the commercially available species that I mentioned previously, and they're exploring more all the time. So this means that they're able to provide a more tailored service depending on the environment, the crop, the pest, and so on, to really give you the best formulation for your needs. And they've now got years of know-how in UK crops to ensure the best performance of these. And I've been working with this when you're deploying Alombria as well, you really need they're really exciting. They've been going for quite a few years now, and they're taking that you've got by technology beyond just the crop that you're working in, environmental so it's not just on the flies that arrive in little boxes. And when to deploy them, so should you deploy them, they also provide any cameras and monitoring services, or should you wait within the crop so these tell you about things like bearing in mind large bonding and their activity levels, their adaptive system quickly, so pest detection, you're more likely to get when those pests are starting a bit more quickly, starting to develop as a problem, and providing wider information. So we've been working with a lot of pollination services, all of the pollinators. So firstly looking at which hoverfly species to use. So there's a whole suite of data that is provided to the grower, three main strawberry web portal species. And these combined commercially can also feed into deployment of lures. So we looked at potato and strawberry aphid, for example, here, if you like. So and potato aphid in a petri dish, proving on developing, and the populations will go up and down if there's no activity. Then these odor lures can be deployed. These are attractive to the hoverflies, so they'll move towards it. And this means that the hoverflies are moved to where they're needed the most, rather than just having to do lots of untargeted releases and hope for the best. So Alombria produce all of their hoverflies within the UK, so I think that's a bit of a unique feature. They have quite a wide species range that goes beyond the commercially available species that I mentioned.
Okay, sorry, just apologies, folks. If you're online, we're also having a problem down in Kent as well. We're just trying to resolve it.
The crop, the pest, and so on, to really give you the best formulation for you.
It's not from, do we know where it
started the second run.
Okay, so I don't know if you can hear, hopefully you can hear me online. Yeah, hopefully you can hear me online. We're having a technical problem down in Kent. So we're just trying to work out, for some reason it was running twice and we don't understand why. We're just going to try and go back to the slide where it all started going wrong, and we'll see if we can run it again.
Thank you, Scott. We can hear you clearly online.
With Alombria to address some of those questions. So it's firstly looking at which hoverfly species to use. And I guess the first question is, are all of the three main strawberry aphid species eaten by commercially available hoverflies? So we looked at potato and strawberry aphid, for example, here. So potato aphid in a petri dish, living on a leaf, the populations will go up and down if there's no other predator added to the system. But if you add a predatory hoverfly larva, then those numbers are dropped down quite dramatically. So you're getting predation of your aphids. And so hoverfly larvae are quite happy eating potato aphid. Strawberry aphid, likewise, put it on a leaf in a petri dish and the numbers will go up and down over time. Add in a hoverfly larva and those numbers are significantly suppressed. So hoverfly is happy to eat strawberry aphid. This is really important because there's currently no commercially available parasitoid of strawberry aphid. So our biocontrols are exclusively dependent on different types of predatory insects, and it's really important that our hoverfly system is willing to go for those strawberry aphid. The consumption rates seem to depend on the size of the aphid. So essentially, bigger aphids, they'll eat more slowly, smaller aphids, they'll eat more per unit at a time. Looking at the two kind of main commercial hoverfly species for pest control, then both of these seem to have similar performance at 25 degrees in terms of the number of aphids that they'll eat in the laboratory. In the field, they're probably both really suitable, really effective, but they'll work in different ways. So exactly where they'll target within the crop, and how they'll move around within the crop. So that precision knowledge is really important to looking at how to release them and combine them, and how exactly we can get the best tune out of them. Previous work from Alombria demonstrates that with correct hoverfly deployments in controlled environment systems, you can get 52% reduction in aphids over 2 weeks just using hoverflies, and that corresponds to a similar kind of increase in the proportion of high quality, so kind of grade 1 and better fruit. So they're just examples of how the two hoverfly species are depressing the number of aphids in those petri dish experiments. So then we wanted to look at when to deploy and how to use lures in order to improve the performance, and do those lures really work? So we were looking in 12 aphid-infested mini tunnels with strawberry crop, and infested those with black bean or strawberry, black bean and strawberry aphid. We kind of threw the kitchen sink at it to try and see what the differences would be with and without hoverflies, and with different sort of deployment strategies. So looked at early versus late, so deploying as soon as the infestation had started to take hold versus leaving it for a few weeks to the point where all agronomists would start to flag that you've got a problem, and looking at whether odor lures help. So we deployed lures at one end or the other of the tunnel and assessed the pest damage and the fruit quality at both ends of the tunnel in each case. So the principle with the odor lures is that if you've got a crop and you've infested some of it with aphids, if there's an odor lure around where that infestation is, it will make the hoverflies more likely to move towards the area, and so hopefully you'll get response faster and you'll get better control. So what did we find? Well, we introduced black bean and strawberry aphid, but this year has been quite something for aphids, and so we did get a few interlopers of species that we didn't introduce but came in from the environment, even though these polytunnels were enclosed and did have closable doors. So everyone's got a lot of aphids. The ends of the tunnels that had lures in had the same number of colonies as the ends without lures, but those colonies were kept down to a smaller level, so fewer aphids per colony. So this we think is quite exciting and worthy of a bit more attention. Equally, when we assessed the fruit at the end of the trial and looked for damage to the fruit, and especially kind of surface molds related to sooty mold arising from honeydew contamination, basically whenever you had a lure, then the level of that damage seemed to be lower than the end of the tunnel that didn't have a lure. So it seems to be helping to reduce the damage level, and likewise introducing hoverflies into the system was definitely associated with a lower level of damage and mouldy fruit overall than no hoverflies at all. Then looking at the quality of the fruit, so just to explain this graph, 4 different categories of fruit, from class 0 to class 3, 0 and 1 being the marketable ones, so the nice ones. Basically, those vertical dark brown bars indicate how much of your total harvest was of marketable quality. And the control at the bottom shows that actually there was quite a lot of misshapen fruit that were coming out this year. Whereas when we deployed hoverflies into the system, there were fewer misshapen fruit, and when there was a lure present, there were even fewer again. So adding hoverflies seems to be associated with a higher percentage of marketable high quality fruit, and adding a lure into the system boosts that further. So how does this translate into recommendations? There's still a lot of work to do over the next couple of years around improving the precision in this system and making sure that we're getting it right in the right formulations for each crop and each pest problem. But hoverflies are showing a lot of promise as an important and helpful part of the biocontrol system. They're suitable for managing strawberry aphid, which is a particular target for us. And both of the main commercial species are suitable for all of the main strawberry affecting aphid species, but knowing how to deploy them to get the best possible performance is key to making sure that you're getting good value for money. Early deployment seems to be helpful, and the odor lures are showing a lot of promise in terms of adding to that hoverfly performance to get the maximum possible benefit. But we'll continue to gather data on this and report back next year, I hope. So acknowledging the funders and the partners on this project, thank you to everyone, and thank you to you, the audience, for putting up with an unfortunately not in-person talk. Please drop me an email, sarah.arnold@niab.com, if you have any questions or comments.
Okay, thank you very much. I do apologise again for the technical difficulties we had there, and it slowed us down a little bit. I'm not going to take questions at this point. Michelle is here, so if anybody has got questions that they want to pick up with Michelle later, please do, or contact Sarah directly. But we are running a little bit late, so we're going to go straight into Francis Wamonje's next talk. Thrips has been a massive problem for the strawberry industry in particular for many, many years. One of the difficulties that agronomists and growers and technologists have is actually trying to identify which species you're dealing with. It's not always very easy at all. Francis Wamonje is going to talk to us about some Innovate UK funded work, which is hopefully going to solve that problem or improve the situation for us. Francis.
Thank you, Scott. So for those who are here during the NAB Agronomist Day, this is sort of like an update of the work so far. But what we really want to achieve is, can we be able to identify thrips to species level in a much more efficient way? But just to give a bit of a background, this is really, I think everybody in the room is familiar with these numbers. Thrips are a serious threat. They cause annual losses in almost up to 50 million by affecting 10 to 15% of cropping. Some of them, such as the Western flower thrips, and for this I'm just talking about those that affect strawberry. They're already resistant to insecticides, and so this always complicates how they are managed. There's a very high diversity of thrips, and especially for those that are on strawberry. So apart from Western flower thrips, you also have rose thrips, you've got onion thrips, you've got flower thrips, and probably a few more are in there. Their size is small, and it's very difficult to identify them. I've watched Celine identify this, and she has to squash a thrips on a slide and clear it, and then key it off. And I think in NAB, there's probably 1.5 people who can do that effectively, the half being Michelle, and Celine being the whole one. And of course, then it becomes very, very difficult to give good advice that can be used for management. So this project started in 2024. We've had 2 phases of it. The first one was a proof of concept. At that point, we thought, let's try and see whether we can develop a molecular detection tool that probably can be used by growers or providers of agronomy services to identify thrips using an isothermal method like LAMP or recombinant polymerase amplification without the need for specialized skills. And we thought we can use Western flower thrips as a proof of concept. It was ambitious, but the whole thinking was, if this works, then we can possibly move this into other thrip species and maybe get industry collaborators to create the detection tools and to make the whole workflow much more friendly to both agronomists and the growers. So we started off by doing fieldwork, and the reason I'm showing this slide is just to show you how the occurrence of thrips species in the field varies a lot. In this particular year, we could hardly get any in town, so we only found 2 samples out of more than 1,600 thrips that were examined. But you could see there was a lot of Western flower thrips, a bit of tabashi, and fishy pennies out there. This year has been different. There have been lots and lots of everything. So again, identifying them becomes something that we need to do. And if we're able to get down to species level, then it makes life much more easier. So what did we do? We started off by looking for the best gene that we could use for species demarcation. And that graph there shows how the different thrip species, their pairwise identity of the genetic information matches up with each other. Out of this, for the first phase, we took the ITS2 gene. We then did a bit of bioinformatics. We aligned the different sequences of the genetic information, picked regions which we saw are a bit diverse, and then began to design primers for that. And then we got 4 candidates. I'm going to move quickly so that you don't see what I have there. But eventually, we were able to come down to 1 primer, which we tested against DNA from mixtures of different thrip species. and we're able to get a specific marker for Western flower thrips. So we're very pleased. And this sort of underpinned the follow-on funding that we got from Innovate UK to then expand this into a 4-species panel. But we got more ambitious. We learned some lessons that the challenge is really, how do we acquire the DNA? If we are going to ask growers or agronomists to, catch the thrips and send them to us, then we've completely we put more work into their hands. How can we make it simple? And so we thought, what if it was possible for us to simply acquire this DNA by doing, flower washes instead, such that a grower or an agronomist can just, pluck 20 flowers, stick them in a bag, send them to us, or if we've managed to develop the technology out there, shake it up, and then get a readout. And we got some very good conversations going with a couple of companies such as Optimising, who are developing some of these systems, and so are quite motivated to go ahead with this project. But the first thing that we had to do was develop then DNA markers that are effective and specific for different thrip species, and that has been the main thing that we've been doing the past few months. But before I go to share that preliminary data, what is environmental DNA? So environmental DNA are traces of genetic material that pests and humans and everybody who is a living organism living in the environment. Right now, where you're sitting, you're shedding DNA. And if we had got specific DNA markers for Scott Raffle, we could come and swap that chair, and we could tell that Scott was seated on that chair if we were that precise. So we're kind of, Scott is not an insect just yet. So we're sort of doing the same thing with the different thrip species by looking at their genetics and finding out, can we get traces of this DNA that can identify a specific species? It makes sampling much more easier because we could get a leaf or a flower, and on there there'll be eggs, there'll be cell debris, there'll be frass. And if we can be able to nail down to specific markers that identify the specific species, then it will make the identification much more easier. And of course, it's able to be done at scale. So the progress so far, we tested 39 primer pairs, which we designed, and some of them which we looked from different sources. 7 primer pairs made the final cut, and they passed. And out of those 7 that passed, we then subjected them. So, okay, let me first show you this gel. So this is an example of what we do. We get what you see as the TTT, FFO, and FI are just abbreviations of the different species of thrips. And we get their DNA, and for each of those markers, we run them against these different species to ensure that that particular DNA marker can only pick one species. And you see, for example, the TTL primer was picking 2 species, and so it's no good. But if you look at, for example, TFUS labeled AFR, it only picks thrips ushipenni, so it's very specific. If you look at the bottom, we've got a specific one for thrips tabashi, labeled B, and C, we do have one for Franklinella, Intonsa. So it's a very iterative, process of designing, redesigning the DNA markers to be able to get one which is specific. But eventually able to get, candidate markers that are specific for the different species. So for those who are here during the agronomist day, I think I'd gotten to 2. Now we have all the 4, so very happy with ourselves, for that. And then we moved on to do, qPCR to see how well these different, markers, do perform. And for 4 of those, we do have, very good performance. So technically, we are at a point whereby we can identify 4 thrip species using molecular markers. So this is what we are on to now. We are now working on getting the DNA from the flowers. And after that, we will develop a full protocol that describes the entire workflow from start to end, such that if we give you this protocol, you can maybe get us flowers, send them to us, or give them to the agronomists. If they're sent to us, then there's predictability as to when you can get your results. But further than that, we're hoping to be able to partner through Innovate with companies which produce this kind of testing devices, so there is scope to have this as field deployable. If you're a grower, you can have your own, or even if you're an agronomy provider, you can have your own device and offer it as an additional service. I'm going to stop right there. And if you come back next year, I'll have more information for you. Thank you.
Thank you very much indeed, Francis. Any quick questions from the floor? One, Robert, if you just wait for the microphone to come down. Just introduce yourself.
Robert Irving. You could identify the thrips. Can you identify the number of thrips per species?
It's not necessary to do so.
Right.
You can. I mean, qPCR is quantitative, and you can have a graph based on the DNA that you have. But in terms of what you will do eventually, is not really dependent on the number of thrips. It's a presence-absence kind of question.
Okay, thank you. We must move on. Thank you very much indeed, Francis. We're now moving on to a Growing Kent Medway funded project, and we've got Damian Flynn from Saga Robotics. Welcome, Damian.
Hello.
I should say this is a Growing Kent Medway funded project. You heard a bit about Growing Kent Medway from Simon Barnes earlier this morning, and it really has been a fantastic project supporting a lot of research in horticulture, but particularly fruit. And this is an example of it. And I'm really pleased that we've got somebody from industry to come and tell us a little bit about what this project has been doing. And it aimed really to develop a way of releasing, I suppose that's the best word to use, releasing predatory mites into strawberries. So Damian, share with us what you found and what Saga Robotics is going to do as a result.
Sure. Hi, can everyone hear me okay? Is that great? First of all, yeah, thank you very much for having me. It's really great to hear all this research going on about the industry moving towards biological means, more sustainable farm practices. I think Saga's been on a mission to remove as much chemicals, fungicides, herbicides, pesticides from the industry as possible. So to get involved in this is really exciting.
I was just waiting for, have you got his presentation?
How does this work?
It's not there. We're just waiting for the presentation. There we go.
If I press this button, does the video start? It does not. Thank you.
Okay, so those of you that are online, we're just again having a little technical problem getting this video working.
Or is there no video? I can probably talk over the video if need be. You could probably mute the audio. That's all right. The music isn't that good anyway. This is Thorvald. Thorvald's an autonomous robotic platform which administers a UVC light treatment to combat things like powdery mildew and other diseases. We've been working hard over the last couple of seasons. This season we did 20% of the UK strawberry harvest. So 1 in 5 strawberries that you ate, British strawberries that you ate this summer, were treated by UVC light. We also did over 1,300 acres of Californian wine grapes as well. Same technology that works in strawberries for mildew, works in other soft fruits as well. So Californian wine grapes are a good industry for that also. But I'm not here to talk about UVC as a technology. Thorvald is particularly well suited because it's in the crop all the time, 2-3 times a week. So when we speak to our customers, they really don't want to buy a single-use robot. They want to buy something that has lots of different functionalities. So actually, if you're in the crop all the time, what the stuff you can do, things like mowing, things like suckering, things like de-leafing. Mite distribution is actually one of the things that our customers said would be really high value. At the moment, they're buying bags of mites and they're distributing them by manual means, I guess. They're opening the bag. I'm pretty convinced that the mites just jump on your face as soon as you open the bag, but apparently they stay in the bag. You then grab them with your hand and you throw them around the crop. And that's not great. It's labor intensive, but also you get kind of clumps of mites everywhere and the distribution isn't very even. So we ran a project to work out, can we distribute the mites via mechanical means? Can we get even coverage? Can we eradicate the labor needed? And can we just make it more reliable? So we came up with a bolt-on tool for the Thorvald platform. So it's we had a number of requirements that we had to fulfill, so we couldn't, because moving a robot in and out of polytiles is actually quite difficult. We couldn't go outside of the mechanical envelope of the robot. So we developed this bolt-on, which kind of sat on the front of the robot. We had a hopper mechanism that the mites got poured into at the beginning of treatment. We had a conveyor belt system that moved the mites from the bottom of the hopper to the top of the robot. And then this distributed the mites onto the top of the strawberry plants, as we saw. There should be a video in the top right as well. Don't know if that's possible to show. You can see it working in action. That's the one, yeah. Possible to play that? No, okay, that's fine. I can talk over it. So this is how I spent my summer, basically, following a robot around, taking videos of mites distribution. So the robot works at night where the UVC works the best, and at the same time, you can just about see the mechanical means on the top there, kind of pouring the mites over the top. And I guess we learned a bunch of things from this, right? I think one of the main things that Niab helped us found out was that the UVC light exposure is particularly damaging to cells, especially things like mildew spores. We were worried that the UVC would also be damaging to the mites as well and hugely reduce the population, but it really didn't. It's such a small dose that actually the light has a very, very small impact on the number of mites. That was great. That was kind of the dream scenario that we wanted. The actual principle of distributing the mites by mechanical means is a very good one, but the actually mechanical implementation wasn't good enough. What we found is that the conveyor belt is very effective at moving the mites from the bottom of the hopper to the top of the conveyor belt with the conveyor system, but the conveyor is also extremely effective at taking moisture from the top of the plants and distributing it into the bottom of the hopper, causing all sorts of problems with moisture and clogging. And we found out you had to service the thing almost every day to get the thing working, which is not suitable for any grower. In fact, one of our customers, my favorite quote of last season, more ag, less tech, please, Damian. Which I don't think he quite meant less tech, but at least could you make this suitable for a farm environment? So I think the other validation that we got is customers really willing to pay for this. They saw the value of it, right? You don't need people, you get much more even distribution. It's just a much more elegant solution for something that's already in the field anyway. So we also got a ton of feedback around how the thing should actually work. So the hopper should be removable or filled from waist height. Currently, you have to climb up a ladder to get to it, and that's problematic if you're having to do it every day. The user needs to be able to see the fill volume as well, so actually getting into the tank was problematic. And we also had lots of interesting discussions around, well, if we see sometimes we see robot problems in the field, whether there's mud or there's something in the way that the robot wasn't expecting. If the dispenser stops working for some reason during operations, do you stop UVC treatment as well? The treatment's kind of interlinked. The robots are there doing UVC treatment. Are the mites less important, or are they as important? Yeah, there's a control mechanism that needs to be sorted out there. But ultimately, like the trial was a success, but the implementation definitely needs to be worked on, and it's something that we're going to continue to do even outside of the funding program. Can we move on to the next slide, please?
For those of you online, we're just trying to move the slide on. The PowerPoint seems to have frozen. There we are.
Okay, so this is our next iteration, right? So one of the things we started to look at is we've had this problem. I imagine other people are going to be having this problem before. And I bumped into a company called Coppert. Oh, I'm sure you're all well aware of, a huge company doing biologicals. It turns out they already have some equipment that is specially suited for biologicals distribution. And it turns out there's a whole bunch of intellectual property around preventing the things that we were seeing, things like clogging. It turns out we weren't just being thick. This was a known problem, and there's actually some smart engineering solutions against it. So in talking to them, they already have a range of solutions for this. The picture in the bottom left is their Naturtech drive. It's something that goes onto the back of a tractor for quite large applications. We wanted something very small that would fit onto our robot. So it turns out they're able to take a bunch of their off-the-shelf equipment and put it together into a custom assembly for us. And they've designed us this, which is going to be our next iteration. So we affectionately call this the Native American tobacco pipe, which effectively does the same thing as our previous implementation. So it has a hopper on the top, but it has this quite unique ball screw design that transports mites from the hopper to almost like a Venturi air blaster, which then takes the mites all the way from the bottom of the robot to the top of the robot via a tube. So that's a much nicer way of doing it. And actually, with Koppert being the experts in the mites and the hardware associated with it, it means that we can focus on the robotics and the kind of platform implementation. There's also very much a supply logistics challenge with supplying mites that have very short shelf lives. So Koppert being able to take care of that for us, and us running customer trials next season with this kind of version 2 is what we're planning to do for next season. Yeah, I think I'll end it there and ask if there's any questions.
Thank you very much indeed, Damian. Any questions? Yes, we have a question.
Hi, Claudia Carvalho, Blue Skies. Question, because mites, they are very sensitive. So going around in all that crushing and the air movement that you need to put them, don't they desihydrate? Always the percentage of that.
So I think Adam might talk about this in his talk later. But I think one of the things that we found was one of the things that we were worried about in distributed by mechanical means, because there's metal and pieces of plastic in the way. So it's definitely something that you need to take into account, the actual delivery mechanism. But we were quite surprised how high the population was. This is one of the work we did with Niab, when they looked at the population, actually putting them in an air in an air channel isn't a problem at all, and they get contained in a vermiculite, like a substrate as well, that has some protection also. There's a few different mechanisms of the market that look like grinders, but they're actually quite gentle in the mite. So we were pretty happy with how well they'll respond to the mechanical means.
Thank you.
Yeah, I know that we've got Jasper Hubert, who's online from Koppert. I don't know if any of your other colleagues are there, but Jasper, you may wish to just, if you've got anything to add, put a comment in the chat and I can read it out later on. If there's no more questions, Damian, thank you so much for sharing your experiences with us. And we're actually going to stick with the same subject for Adam Walker. So Adam is another entomologist in Michelle Fountain's Pest and Pathogen Ecology team here at Niab's East Malling site. Niab worked with Saga Robotics on this project, and in addition to the actual logistical mechanism for releasing mites into the crop, we learned quite a lot more about predatory mites and the timing of introduction. And Adam is actually going to share some of the findings and learnings that he has picked up from that work. So Adam, share that information with us, please.
Thanks, Scott. And thanks everyone for joining this presentation. So today I'm going to be sharing information about optimizing the timing of predatory mite release in strawberry. Now, there are a number of factors which influence the timing of predatory mite release. Things like the presence of their prey, presence of alternative food sources, and when pesticides are being applied. But the talk today is focusing on the two main influences, which are temperature and humidity. And what we've done for this presentation is we've reviewed manufacturers' guidelines and also the literature for 4 species of predatory mites, which are typically applied in soft fruit crops, including strawberry. And then we've assessed them on the effects of temperature and humidity on 4 key life characteristics. Starting with general activity, so that will be their hunting, catching the prey, feeding, reproducing. Predation, so we know at optimal predation ranges, they're going to catch more prey, they're going to feed on more prey. Reproduction, so this is obviously egg laying. We know that at high temperatures and dry conditions, Eggs laid tend to dry out and not hatch. And life cycle and longevity, we know also at high temperatures, they'll go from egg to adult more quickly, but it can also reduce down the longevity of those adults. So the way that I've chosen to present this information is in a series of graphs, first focusing on the effects of temperature on the 4 species of mite, and then humidity on the 4 species of mite. So starting with Amblyseius andersoni, I'll explain the graph. The fluctuating line in the background, that shows the daily fluctuations in temperature from March to October recorded in a commercial strawberry polytunnel. And then overlaid on that, the faint coloured band, that shows the range of temperature that Amblyseius andersoni are active. So in this case, 6 to 40 degrees, it's a big range of activity for this predatory mite. But then this darker coloured band in the middle, this represents when this predator is most predative. And you can see it's a bit narrower here, so it's from 20 to 28 degrees C for predation for Amblyseius andersoni. And it's worth noting that generally not many eggs are laid below 17 degrees C. So same format for Amblyseius andersoni, cucumeris. You can see the active range is from 15 to 30 degrees, but the optimal predation occurs between 15 and 25 degrees. And this predator tends to not be very effective above 30 degrees. So you might want to use something else for thrips control if you get kind of sustainable temperatures that range. Which brings us on to something like Amblyseius swersky. So the temperature range that it's active is 20 to about 32 degrees, but optimal predation occurs between 25 and 29 degrees. And this predator tends to be not very effective below 18 degrees. So it's more effective at those kind of warmer, drier conditions. And finally, Phytoceus persimilis. So it's both active and its optimal predation occurs between about 13 and 27 degrees C. It tends to not be very effective above 30 degrees C, and at 35 degrees C, it tends to stop feeding altogether. So you might want to consider alternative spider mite control under sustained conditions like that. So same format, but moving on to the humidity ranges of these predators and back to Amblyseius andersoni. So in this case, well, in fact, what I'll say first of all, what you'll notice about these graphs is all of these predatory mites tend to be more effective at higher humidity ranges. So that's really important for that. They're very susceptible to drying out. So drier conditions are unsuitable for these predatory mites. But back to Amblyseius andersoni. So it's the range that it's active is 60 to 90% relative humidity. But optimal predation tends to occur above 65% relative humidity. You'll tend to get high mortality below 50%. Amblyseius cucumeris. So it's active from 65 to about 85%, but optimal predation occurs between about 65 to 75%. There will be a reduction in egg hatch below 60% relative humidity. Amblyseius swersky. So again, this, like I said before, this can tolerate dry conditions. So it's active between 55 and 85%, and optimal predation occurs above 60%. You'll tend to get a lower fecundity and egg hatch below 50% though. And finally, Phytoceus persimilis. So it tends to be active and optimal predation occurs between 60 and about 90% relative humidity. Egg hatch is reduced below 60%, and you get very little to no egg hatching below 50%. So that's the kind of seasonal effects of temperature and humidity on these 4 species of predatory mites. But we've also got to consider the daily fluctuations that we might experience as well. So the graph on the left-hand side is dealing with degrees Celsius, and this shows the 24-hour fluctuation in degrees Celsius in a strawberry polytunnel. And I've chosen August as an example for this. So you can see it starts off at midnight at about 15 degrees. It peaks at about 32, 33 degrees around 3, 4 o'clock in the afternoon. And then by 11 o'clock in the evening, it goes down to about 15 degrees. So these temperature ranges are actually okay for all of the species that I've just talked about. But if we move on to the right-hand side, this shows the humidity ranges of these predatory mites. And you can see in the nighttime, so from midnight till about 10 in the morning, it's above 65% relative humidity, which is suitable for these mites. But then from 10 to around 6, it dips down to kind of more critical levels of humidity for them before rising up again. So this suggests that releasing these mites early in the morning and in the evening are better, at least for them to settle without getting dried out. So it's better to release them at those times rather than the middle of the day in August. But you've got to consider the temperature and humidity fluctuations throughout the year when you're making those decisions. Now, a word on our work with Saga then. So we looked at the effect of the UV treatment that Saga use alongside how it affects the survival of Amblyseius cucumeris released by their dispensing device. And what we found is there was no obvious impact of the UV treatment on the survival of Amblyseius cucumeris. Also, there's the potential advantage, so as I've just discussed, in August or during the summer months when a loose formulation would be released, is that nighttime application allows those mites deployed on the crop to settle better. So to conclude then, for preventative control, apply andisoni as sachets, usually in rubus species, when average temperatures are at least 10 degrees C. And Amblyseius cucumeris also sachets from about 15 degrees C. Then loose formulation can be applied when the thrips appear. Persimilis is effective from 13 to 27 degrees C. Remember, that's a specialist spider mite predator, but it's not so effective above 30 degrees C. So long as there's not webbing, something like Swirsky can be put in to help control spider mite, which is effective at those warmer, drier conditions. And it's important to time predatory mite releases according to daily temperature and humidity, so keep monitoring those. And also, of course, keep monitoring the establishment of your predatory mites in the crop as well. So look for eggs, look for juvenile stages as well. Obviously, monitor for the pests as well and the control of those. And I'll give an example. So early in the summer, early in the morning, evening applications, as I've discussed, is potentially better because of the high humidity favouring the mites settling. And as I mentioned, Saga's UV treatment did not harm mite survival. Also, the nighttime application is potentially more favourable for the mite settling. Okay, that's it.
Thank you, Adam.
Sorry.
One more slide.
A couple more things. Forgot about this. I've provided these tables, which will be available with the presentation. So this is the information, a summary of the information that I've just given. A couple of things I didn't go into so much detail on. So I talked about the active range and the predative range of these predatory mites at different temperatures. This also includes the reproductive range and the life cycle and longevity of them. And I've done the same for humidity as well, so you can refer to these.
I'm sure that'll be very gratefully received by all agronomists out there. Thank you, Adam. Right, we've got a number of questions that have come in from the chat, which actually relate to both of these presentations. So if Damian can be on the standby. Any questions from the floor initially? If not, I'm going to go straight into the questions online. So Michelle, if you can probably put this one to Damian. So if you want to take Damian's over there, if you want to take the microphone over there. The question is from Delilah Burnstone. What about the treated area? Growers, if they're using other biodispensers, they will treat 6 lines at one time. With the robot, you can only treat 1 line at a time. Can you adapt the rate of bio release to the speed of the machine?
Yeah, I mean, you're right. You can only fit the biological distribution means through the capability of the robot. The robot does a single row for a reason, because it's really hard to move autonomously in polytunnels, and it's the edges which are the most challenging parts. The mite distribution is already linked to the robot's speed. So when the robot stopped or when it's moving slower, you're not over distributing or under distributing. Is there plans for a multi-row robot? No, not at the moment. Yeah, I think the challenge was to fit the mite distribution into the mechanical architecture of the robot, which is only single row.
Thank you. And hold on to the microphone, because another one for you. I think you've answered part of this question. When is the mite dispenser going to be commercially available? And what about the de-leafing and mowing options mentioned earlier in your presentation?
Yeah, I mean, there's ideally, I'd have a huge team which will be working on all of these operations with the robot. Unfortunately, we don't, and we've kind of fit in our means. So we have to kind of prioritize. As additional bolt-ons to UVC, we prioritized data last season. So we offer a data product. We're capturing fruit counts and brightness assessments. Mite distribution was kind of the second highest priority because of customer feedback. It's not commercially available yet. We're hoping to do a trial with the kind of version 2 next season. But if that goes well, then I guess we would hope to offer it commercially the season afterwards.
Okay, thank you. And Delilah's come back with another further question. In the spring, will the release of predators be more favourable during the day because of the low night temperatures? Don't know if that's a question maybe for Adam.
Yeah, oh, sorry, I don't need the mic. So yeah, spring humidity, it tends to be higher. So it would be suitable during the daytime. Plus the temperature ranges are probably more favourable for these predatory mites to be active. So yeah, I would say it would be okay during that time of year, but obviously keep monitoring that. And when you get into those warmer, drier periods, be prepared for early morning and evening releases. I mean, I'd be more inclined to go for evening releases, because then you've got more time for the mites to settle. They might even do some predation and lay some eggs in the process.
Just another comment in the chat here from Stephanus Mulherb, who I think is based in biological services in Australia, if I'm correct. Just a comment, entomatic MRD can treat multiple rows in one pass for predatory mite release, and he's put a link to that in the chat. So for those of you who are on the chat, that you can have a look at that link. One last question before we break for coffee, and this is possibly a question for Michelle. In the absence of Sarah Arnold, can hoverflies be used in vegetable production?
That is a good question, and I'm not aware of what research is going on with hoverflies for vegetable production, but yeah, they're quite generalist in the aphid species that they eat, so there is an opportunity there to work on that as a biological control.
Okay, that was a question from Darlington Tenkerang. Any final questions from the floor here in Kent? No. So we're going to kick up. We've got 4 talks now between now and lunch. We're going to start off with Michelle Fountain, who's going to talk about parasitoids for SWD control. Francis is back on the stage for the 3rd and final time, I think, talking about brown marmorated stink bug. Then we've got Michelle back on talking about some projects she's been doing on AgroBioConnect and the landscape diversity around strawberry crops. And then the one and only storage project that we have of the morning will be Richard Cogan from the University of Greenwich. So I think everybody is well acquainted with Michelle Fountain. She's been talking on these platforms for many, many years. Michelle heads up the pest and pathogen ecology team here at Niab's East Morningside. Michelle, tell us all about your work on, this is another Growing Kent Medway funded project in association with WB Chambers and the British Berry Growers Association. And it's looking at parasitoids for SWD.
Thank you, Scott. And yeah, so you're all really familiar with SWD, so I'm not going to introduce SWD, spotted wing Drosophila. So I'm just going to talk about the parasitoids today. But none of this work would have been possible. So I don't actually do any of the real work. So it's the team that do the real work. So Adam orchestrates everything. Francesca's been really involved in the SWD rearing. Selena is our taxonomic expert. So she's been identifying all these things that are about 1 millimetre long. And then Adrian Harris has been helping us in the development of an augmentorium for parasitoids. So just to remind you that encouraging sort of biological controls, in this case, parasitoids, could offer a low input control strategy for Drosophila suzuki. The parasitoids we're talking about belong to the Hymenopteran order, so they're related to bees, ants, and wasps, and they lay the difference being that these lay their eggs inside or on a host, and then the larva, when it hatches, kills the host by feeding on it. And if you remember from way back when we first started talking about SWD, it has this really clever way of closing the parasitoid egg inside its body so the egg can't develop. So that's why it's really tricky to control normally with parasitoids. There is a species called Trichopria drosophili, which is in Europe and is commercially available for release. But because we haven't identified this species in the UK, we're not allowed to release that at the moment. But we do have some UK native generalist parasitoids, which I'll talk about. So this was some work that the team did between 2017 and 2019. There'll be some names there that you remember, that you will recognise, that are in the industry. And we identified in the UK 4 pupal parasitoids and 2 larval parasitoids. And the larval parasitoids are pretty good. And just note the one at the bottom there called Leptopolina heterotoma. So that's a native species to the UK. Also in this work, we found that their activity is somewhere between May and October, peaking in the middle of the season. And we mostly find them in semi-natural habitats. And where we did some field testing of the control that these parasitoids can give you, we get about 21% reduction in emergence of SWD adults where we have parasitoids. And the one that I'm going to talk about a little bit today is Pachycropoides vindemi. Scientific names, I don't know whether I'm saying that correctly, but that's how I pronounce it. And we also found that this species in the lab could increase its population size reared on SWD. So that's really interesting. So the projects I'm going to talk about today, the first one was a Growing Kent Medway project, which was led by British berry growers. And then I've got 2 exciting developments that have happened in the last 12 months to tell you about. And then I'm going to finish off with a British berry grower funded project and give you an update on that. It's still ongoing, so I won't be able to give you all the results, but I can give you a taster. So firstly, the Growing Kent Medway project. So here we were trying to develop an augmentorium. And what I mean by that is something that is self-propagating. So a device that we can put onto farms that helps propagate parasitoids to help you control SWD. So I can't show you what it looks like just in case we want to protect it. But basically we put them into these boxes in the middle of woodlands, because remember SWD is in woodlands from the autumn right through to the spring. That's where it overwinters. And so we want to get a good hold of it there. Parasitoids don't survive very well inside cropping areas, so we target the semi-natural habitats around the crops. So inside our augmentorium, we put waste fruit from the farm and we inoculate the waste fruit with Drosophila melanogaster. So we're not releasing Suzuki on the farm, we're releasing melanogaster into these augmentorium. And we know that our native parasitoids like to lay eggs in melanogaster. So we're propagating it within this system. Then to test how it works, we had these sentinel traps which contain fruit with SWD in, and we had grids on. Some of these traps will allow parasitoids in and out, and other ones will not allow parasitoids in and out. And then we can look at the difference between those two trapping systems, and that will give us a percentage of control of SWD. So all of that explanation for this one graph from the Growing Kent Medway project, and you can see where we didn't allow parasitoids in boxes, which is the orange line. For most of the season, it was fairly similar to where we did allow parasitoids in and out of the boxes until the end of the season, when we started to get a little hint that there's something interesting going on. It wasn't statistically significant, but it gave us a bit of enthusiasm to carry on with this work. Also, in the Growing Kent Medway project, we looked at the species of parasitoids that were coming out of both the sentinel traps and the waste fruit, which was pretty gross actually for the team to deal with, as you can see in the bottom right-hand picture. And interestingly, we didn't get any Pachycropoides vindemi in that year, but we did get 4 species, and super interestingly, we got a species called Leptopollina japonica, and the clue is in the name, and I'll come on to that in a moment. So because we got Leptopollina japonica, this is a native of Asia, and it's more specific to SWD. It does also lay eggs in Melanogaster and Drosophila similans as well, so 3 species that it targets. But it's really good news for the industry that we found that, because it means it's here and hopefully it will establish. So Harriet Duncalf, who you all know, working on behalf of British berry growers, worked hard with Neil Audsley to get this through with DEFRA so that we can now release Pachycropoides vindemi onto farms commercially. And there's a company there called Katz Biotech, who are based in Germany, who are producing that species at the moment. So remember, we don't have the ability to release other species that the rest of Europe and US, etc. do. And then Glenn Powell, who you might recognise the name, used to work with us. So he was doing a bit of a bio blitz in his garden, and he also found Leptopollina japonica in his garden. Actually, I got it the wrong way around. He found it in his garden first, and then he told us he'd found it. So we then went and checked our samples, and we'd got Leptopollina japonica as well. So he's based down in Ashford, and the one we found was in never eat shredded wheat, West Kent. Thank you. And the way you tell the difference between Leptopollina japonica and Leptopollina heterotoma, which is our native, is if you look at the bottom right-hand picture with the letter C, it's literally a slight change in the shape of that sort of raindrop shape on the back of the parasitoid thorax. So we have 2 options now then. So we can look at Pachycropoides vindemi, which is our native species, which is now commercially available for inundative releases, the same as we would with, for example, Ambuloceus cucumeris for thrips control. So it's a generalist species. It has a lot of hosts. It's not particularly fussy. And to add on top of that, it's also a hyperparasitoid as well. So it can also lay its eggs inside parasitoid eggs that are already in a host. The other thing it does do, which is interesting, is it injects venom into the host when it's doing its kind of probing and feeding. And I think that's why we get a higher kill than the number of parasitoids that emerge from SWD, because there's some killing going on with its venom. And it also can regulate the host immunity. So it helps stop this eclosing of the eggs. So that's really, really important for SWD. Really ubiquitous, found in 60 countries, and we think it probably overwinters as a larva or a pupa in a wide range of habitats. So it's obviously using other species to overwinter. And then the second one, the non-native one, which is Leptopollina japonica. There are about three species. Actually, Ganapsis has changed its name again. I think it's Comorum now. But these are the species from the home range, which are really effective. And Leptopollina japonica, which we now have in the UK, has also been found in Sicily, Canada, Italy, Germany, North America. So it's called adventive arrivals. So they're arriving of their own accord, probably with fruits that have SWD in. And in other research, in areas where it has naturally arrived, it's about 20% control of SWD, but it can range from 0 to 70. And this is particularly dependent on the environmental conditions. So then then British Berry Growers funded the next tranche of research, again with the Augmentorium. So this time we had woodlands with Augmentoria and woodlands without Augmentoria. And we inoculated the Augmentoria waste fruit and Melanogaster, and we put our sentinel traps out in the treated and the untreated woodlands. But this time, of course, because we've got Pachycropoides vindemi available, that meant that we could also boost the local populations of parasitoids with this species that's now commercially available. It takes 6 weeks for the so even though SWD life cycle is every 2 weeks, the parasitoids is a 6-week cycle. So it takes a long time for us to rear them out of the pupae of SWD. So we're still collecting that data in, and Celine is going through all the samples and identifying these minuscule little parasitoids. So I hope at the next meeting we can present that data. One of the other things, though, that British Berry Growers did fund as part of this project was a survey of where Leptopolina japonica, the non-native species, is in the UK. And I just want to say thanks to all the growers who have helped us with this, because many of you have been out collecting wild blackberries, and James Hutton also collected blackberries for us in Scotland. So we looked at Kent, the West Midlands, Scotland and Eastern England. And we took 2 samples from each farm, and they're about 3 or 4 weeks apart. And again, we incubated those for 6 weeks. And Selina's working her way through all the samples, doing the taxonomic identification. If you look at the picture on the top right-hand side, you can see a couple of SWD getting romantic on that blackberry. And then the picture below that, you can see there's a little parasitoid sitting on the edge of the pot where the blackberries are being collected. So they're quite keen. So this is sort of hot off the press data. And the thing that we can see here is that Leptopolina japonica was found in the 2 southeast sites. And actually, we have also identified it from Francis's work on the raspberry as well. And that's also southeast as well. So it seems like it, might be taking hold in the southeast. Now, the other big orange red bar in the middle, you can see, we caught lots of Trichopria species from a site in East Anglia. Selina is working through the identification of those with a taxonomic expert, because there's no key for keying them out to Trichopria drosophili. In the past, we have found other Trichopria species, but we don't know whether this is the drosophili species yet. So that information, hopefully, we'll get that to you soon. And that's my talk. I just want to say thank you to the host growers for helping us and collecting blackberries, and also to the team who made the work possible, and Berry Gardens and Growing Kent Medway for funding. Thank you. Sorry, not Berry Gardens, British Berry Growers. Too many similar names.
Thank you very much, Michelle. Any questions from Michelle, either online or in the room? One quick one from me is, what's the situation with Leptopolina japonica? And, if it is appearing more in the UK, at what stage might it become available for release?
Well, it would still need registration. It would need to go through Natural England. I suspect it might not be available for commercial release, because it also lays eggs in Melanogaster and Similans, non-targets. But Napsis camorum is more species specific and is being used in other parts of the world for control, and is pretty good. So that's maybe something we should.
Okay, very clear answer. It's not going to happen in the short term.
No.
Okay. Louise, yeah, we'll just wait for the mic. There we go.
Thank you. Louise Sutherland, Chair of the British Berry Growers R&D Board. First, thank you very much to everybody who's been involved. I would like to say to everybody, I know it's really exciting news and you might want to rush out and buy a whole load of parasitoids, but we are doing some more work this year in terms of optimising those conditions. So we hope to be, by the end of the year, to be able to tell you some of those tricks and tips to get them to work.
Wonderful. Thank you, Louise. Okay, let's move on. Thank you very much, Michelle. Michelle gets a rest, and she's coming back to join us again shortly. In the meantime, third and final time for today, Frances Wamonje is back. Now, Frances has been doing some work on brown marmorated stink bug, and many of you may think that brown marmorated stink bug is a bigger issue for the apple and pear industry, but it does cause damage to soft fruit crops. And so for that reason, we felt it was appropriate to include a short update from Frances. Frances is being funded by DEFRA. We're very grateful to DEFRA because they continue to fund surveillance work for invasive pests. And Frances is going to give us an update on what that work has been yielding. Frances.
Thank you, Scott. Just maybe to add, aside from DEFRA funding the main surveillance, there's also been traps which have been bought by British berry growers and British Apples and Pears Limited. So I would like to thank industry as well for participating in this. But yeah, that's the outline of the presentation. And I'd like to start here just because one of the main ways in which we get information about the occurrences of brown marmorated stink bug is through email. And so we get lots of pictures sent to us via that email, and we go through all of them and we give feedback. Sometimes, yes, it is a BMSB, at times it's not, but we usually will send you a picture of the distinguishing features so that identification can be much more accurate. And just the other thing is that DEFRA have updated their BMSB fact sheet, so it's much more recent and much more updated based on the information, the feedback that we've been giving. So that's a link to it. So BMSB, as Scott had mentioned earlier on, is highly polyphagous. It will basically infest a whole host of crops. Doesn't seem to pick at all. But these are USA pictures. This is not in the UK. As you can see from these pictures, it can also affect soft fruit quite well. And you can see this causes all manner of damage. Most of it is physical, but there's also physiological, reprogramming, as you can see with the blueberry picture on the right, whereby it causes ripening to occur prematurely. But it also has metabolic, problems, especially in strawberry, which causes the fruit to have a very distinct odor, and you cannot sell that to anyone. So it's potentially a very damaging, pest. And it can also be a significant, public nuisance. So this is usually the picture in parts of continental Europe and Turkey. Whenever it gets to the autumn, when these begin to they'd like to overwinter indoors. So they will look for any place where they can get in. This will be barns. We had contact with a gentleman from Turkey and said the mosques are full of this BMSB as well. So we really don't want to get to this stage with this pest. This is the latest global map of where BMSB is established. So in as much as the UK has had regular interception since 2018, when records began to be kept, it's not yet established in the UK as yet, but it seems to be probably a question of when, not if. And this is a picture showing modeling that was done a couple of years in 2021 to show the likelihood or the areas which would be most likely to be affected by BMSB by 2050 should the temperature increase continue. And I think I'm aware that there is some suggestions of work to be done on modeling for the kind of temperatures that would be within semi-controlled environments and what that would look. So that probably would be much more accurate than this. I don't know whether Glenn is involved in it, but we certainly look forward to seeing that the results of that climate, that modeling. Now, this Climex model was used to identify the initial sites where we have been having the traps going since 2021. And DEFRA have been very kind to, to be funding that. And so this is what the picture looks like. In the UK, since 2010, there have been interceptions, and Niab has been involved in monitoring since 2018. In 2021, the first female BMSB was identified in the UK, rather reported to us. From 23-24, and this is largely through partnership with all of you and some of you who do the monitoring, for us, we've noticed an increase in incursions where more than one insect is involved. And last year we had the first report of juvenile forms in the UK. Now, one of them was predated on video as we watched by a local spider, but the other one was, I think, reported to DEFRA, and some control measures were taken. So if you look at the numbers, initially we're getting reports of single occurrences, but then we began to see much larger numbers because, for example, you'd have a truck coming in with some imported goods, and they're conscientious and they report to us that this truck has got over 100 insects. We send them traps, they kill as many as they can. Some of them would engage the services of a fumigator or something of the sort, but the numbers do go in. And from this, we've been able to identify that this is how these incursions are actually occurring, mostly through trade and through leisure in camper vans. And if you look at 2024, the purple at the very top, those are the two records that we had of nymphs occurring. So far, 25, until yesterday, we've had 30 reports. A couple of those reports involve more than one insect. So that seems to be the ongoing trend that is going on with BMSB in this country. And we're getting a lot of support from Harriet, who's chairing our the invasives committee on BMSB, very closely following up on these numbers and to see what kind of actions can be taken, what kind of lobbying may be needed to get us better prepared. In terms of the options for monitoring and control, pheromone traps, the evidence is clear, they're quite effective, and that's a picture from Glenn, having caught one when he was still at RHS. In fact, this year, we have a different strategy this year. On top of having the sentinel traps, whenever anybody writes to us and says, I've seen a BMSB and we confirm that it's one, we do send them a trap. And on at least 2 or so occasions, those traps have caught additional BMSB, or the person has found additional BMSB within that area, and they do send them to us. We are keeping this BMSB because we are also interested in the genetics of this insect, and we've got a small pocket of funds, likely from Shaming, to have a look at this as a side project between January and March, just to try to get understanding where are these incursions coming from. But that's a science part. In terms of what other people are doing is to try and find different ways in which you can make traps more effective by making them bimodal. For example, this paper showing an Nazgul trap, and that's essentially a net which is impregnated with insecticide, and it works in conjunction with the pheromone trap, which is really very, very effective, so that you can have more or less like an attract and kill strategy for this. But this really would be the best way to do it. Trisochus japonicus is a very effective parasitoid of BMSB. There have been adventive populations found in the US and on continental Europe. It's been deployed in Switzerland, and in Italy, it's been quite effective. My apologies, my wording seems to have slipped out of place over there. And in New Zealand, where BMSB is not yet established, but like in the UK, they do have incursions which get intercepted. They have pre-approved the release of Trisochus japonicus. And in the BMSB working group, we're kind of keeping the conversation going to see whether it may be possible to have a similar approach, because wherever it's been deployed, it's been really, really effective. But the main thing which we are doing is outreach, because we need more and more people reporting to us whenever these are found. We got fantastic buy-in from the Camper Van and Motorhome Owners Association. We created this infographic, largely the work of Scott and Michelle, having a very nice back and forth. It's nice and yellow and bright. And this went into their online, it went into their print. And after that, we've always had people reporting back to us, telling us whenever they find BMSB. I think just 2 weeks ago, I went down to the Bearsted Motorhome and Camper Van pack to get some BMSB because somebody had seen this, recognized it, and got back to us and said, hey, I've got 4 of these insects. Can you come and pick them up? And so this is the kind of feedback which we are getting from people, and it's working really well. In additional work, and this is funded by the Worship Company of the Food Trust, we are also interested in what could be the native parasitoids that could be used in the biocontrol or biotesting pack. So in this experiment, what we're doing this here is rearing these insects and collecting the eggs. We kill them by freezing, and then this will be deployed out there for a predetermined period of time. We will retrieve this and then see which parasitoids emerge from this, but also try and keep an eye what could be predating on these eggs. So we're trying to prepare for a future that may occur by getting a whole collection of things which we can be able to use in the control. Thank you.
Thank you very much indeed, Francis. I just a little bit of point of information. Harriet Duncaff, who's joined us online today, she chairs the Spotted Wing Drosophila Working Group, and she has done for many years. I act as secretariat to that group. And in recent times, she has just set up a brown marmorated stink bug working group. So that was what Francis was referring to, which has a whole range of organisations are represented in that, including British Apples and Pears Limited and British Berry Growers Association. So it's very much industry involved. Any questions to Francis? Yeah, we've got one from Jonathan. Thank you, Jonathan.
Yeah, Jonathan Blackman from Hutchinson's. Just to clarify, did I understand you said that you've got some money to look at genetics, so you'll be looking at the different halotypes and what you're catching?
Yeah, it's just a small part of money that we have. These samples which we collect and which people send to us, we've been very, very deliberate in getting them in. We think that if we look at the genetics, we can probably understand where the incursions are coming in from. So we'll see what the preliminary data looks like. Maybe by June, July, I'll be able to give an update on what that looks like.
Because it's known that one of the halotypes has a lower degree day requirement to complete its life cycle, and obviously if that was the one that we were getting here, then the chances of it getting established would be much higher than if it was one of the others.
Yes, we definitely want to look into find it. Yeah.
Any more questions? Just another one from me then. Trisochus japonicus is obviously quite active in Switzerland or being used in Switzerland. And do you want to say anything about the work that's going on? Because there's a little bit of work going on here, because we're looking at a possibility of potentially getting its use authorized at some point in the future.
So the best data set is actually from a group in Italy. And Italy has had a couple of trials where they have released those, and you can see the numbers really coming down. But also the Trisochus japonicus establishes really, really well. So it's been very, very effective. For Switzerland, in fact, that was Tim Hay, who has been our main collaborator in terms of giving us technical advice. And he just mentioned that previously, every year he could go out there and get as many BMSB as he wanted for his experiments. But since Trisochus japonicus was released, it's become harder and harder for him to even find material to work with in experiments. So they're quite effective wherever they're released.
Okay. Thank you, Francis. We'll move on. Thank you very much. And you can have a very good, well-earned rest now for the rest of the day. Thank you for all your hard work and effort. Okay, we're moving back to Michelle again. Now, Michelle has been working on a project which DEFRA has part-funded, and an EU project called AgroBioConnect, which has been investigating landscape diversity on pest control in strawberry. And she's going to share some results. That work is ongoing, I believe, but you're going to actually share some results of a survey you've been doing with growers as part of that project. So, Michelle, tell us more.
Thank you. So, yeah, apologies, the title changed because I just looked at the amount of data that was coming in from Francis and Sarah and the team, and I thought I haven't got time to get my head around it for this meeting. So again, hopefully we can present some of the outcomes of that, because there's been lots of molecular work, lots of taxonomic work that's coming through. So as part of the project, there's also a social science aspect. But just to give you a little bit of background for the project, so obviously we have a growing global demand for food. And this has resulted in intensification of agricultural practices and simplification of agricultural landscapes. So this project is all about landscapes and how they impact ecosystem services. And intensification can obviously lead to ecosystem degradation and a loss of biodiversity, which we hear a lot about. And it would be ideal, obviously, to transition towards a more agrobiodiversity-based practice on farms. So the actual kind of practicalities of the project is we're looking at the relationship between landscape complexity, and that's an image from the Sentinel-2 satellite, and we're analysing, not we, sorry, the partners in Italy are analysing that data in with our biological data that we're collecting on the ground. So we're looking at soil biodiversity, but also in the UK, we're really focusing on aphid parasitoids and pollinators in strawberry crops. So that practical work has taken place over 18 farms over the last 2 years. And for the survey, I got agreement from 7 of the 11 strawberry growers to survey them about their opinions on landscapes and how that interacts with the crops. And you can see that the growers that I interviewed, they also grow a range of other crops, and their roles on the farms also differ. So hopefully we're getting a sort of a reasonable range of views of what happens. For the interviews, I used Teams decisions to record the interviews, and then I went back and cross-checked that Teams had got it right. And then I also used Copilot to help me write the report, which we're hoping will be in the Fruit magazine, looking at Scott, in the next edition. So we'll get the full report out to you. And I asked the growers 5 questions, and the first question was, how much do the strawberry growers consider the wider landscape in which their farm sits and how that influences the farm? So some of you were in here as well, I think, and probably online, but everything is anonymised. So one of the first things growers talked about was older fields and the fact that their fields have been established over a long time. So some of the field shapes and sizes have been established for decades and decades and are difficult to change. They also talked about that their attitudes are changing, and that when they design new fields of strawberries, that they're actually they're doing this with biodiversity and habitat management in mind. In particular, they're interested in diversifying flowering hedgerows and reducing mowing on their farms. I saw this paper from last year, I think, and this is really interesting. So this is improving and increasing semi-natural habitats in organic strawberry farms in California. Hence, the birds all look very pretty. And what they did by doing that is they tipped the balance in birds that ate capsids, as opposed to birds that ate strawberries. So by diversifying the food available for birds, they actually tip the balance in favour of capsid-eating birds. The growers also talked about their worries about the negative influences of landscape on pest management, and how previously they'd been more worried about the landscape as a source of pests and diseases, but that their views were changing, and they really want to work with these habitats to sort of reduce the numbers of pests and disease in the landscape, but also how can they manipulate them to encourage and boost more beneficial insects. Probably unsurprisingly, water came up as a big concern about the landscape, particularly with more hard landscaping. So you have water shortages in the summer, followed by flooding in the winter, and the need also to recycle the water that's being used on the farm for those particular dry periods, and being able to manage water runoff and manage the water that's coming off those catchment areas. One of the growers is also located in one of the national parks. So he was talking about the need to be extra careful about the sensitivity of wildlife in that area. Then the second question I asked the growers to do was to rank, and this has been done in Europe as well, so they're also interviewing their farmers and growers, is to rank 6 landscape factors in order of importance in terms of diversity and complexity. So we were comparing worst and best. So for example, a field size, the worst for diversity complexity would be large field sizes, field edges, if the worst scenario would be straight as opposed to sinuous field edges, connectedness as well. So here we're talking about corridors so that things can move around the farm. And I think I've mentioned this in the past, that there's no point in having all your habitat at one end of the farm in one big field, because how is it going to distribute across the rest of the farm? We talked about habitat types and which, whether they were important or not, and also the quality of those habitats. So we could take a poor quality habitat might be strawberries in polytunnels, and then we compare that to flower-rich meadows and woodlands, which would be high quality. And then I also asked them to think about the landscape as a whole and how important they think that is. So those are the answers from the individual growers that I spoke to. And what I did is I took the average of their answers, and you can see that habitat connectedness and corridors, the growers considered the most important things in the landscape on their farms, and that was to facilitate the movement of natural enemies and wildlife and pollinators. And then second came habitat quality, in particular of woodlands and flower-rich meadows. And then after that, habitat type, although one grower you can see on that top graph ranked that the highest. And that habitat type included things like edges, but it was kind of mixed rankings really for that. And then field size came up, particularly with one grower who suggested that in his smaller fields, he gets fewer problems with aphid control because there's more edges, but potentially more natural enemies coming in to control those aphids. Other things that we talked about under this were having dedicated areas for habitats and space for biodiversity and nature, and the problems of practical implementation of doing this. And surprisingly, thinking about the bigger picture, the overall landscape complexity of diversity scored really low. Growers are really, really targeting those areas that they could actually have some control over. And then in the third question, I asked, how do you think landscape benefits your business? So landscape, there was a lot of talk about hedgerows and windbreaks and supporting wildlife, insects and birds. And I've tried to put in some websites through this talk, by the way, that you can visit to find out more information. So farm wildlife is a really good one. And I really like that picture on the top right because it has mixed flowering hedgerow, and then it has a canopy that comes down to the headlands. So it's providing in that space lots of different habitat for insects and birds, etc. Growers also talked about the provision of ponds, but they weren't really sure what they were providing. But ponds provide obviously water for animals, birds as well, and also they're quite important for pollinating insects. So pollinating insects need to be able to access water. But they did say that they don't have any information. They can't quantify what all of this is giving to their fruit crops, and that can be a bit of a barrier. And there's still a really heavy reliance on purchased biological control. And this is seen as also really important for helping to produce residue-free crops, which is demanded by retailers. One of the conversations I had, I really enjoyed, obviously, because it was all about wildlife and nature. So one of the growers was really enthusiastic that he was seeing the return of wildlife to his farm by putting some of these practices in place. And there's some of the animals that he mentioned in the conversation that have returned to his farm as a consequence of changing management, and the fact that he got personal satisfaction from that. And that promoting biodiversity is quite often separate from farm profitability. So it's seen as the right thing to do. But what that does mean is that it's a risk to biodiversity because of extreme financial pressures that farms are under. It does put that biodiversity at a bit of a risk. Do I grow a crop or do I put a habitat in? And again, those economic gains were difficult to quantify. And who bears the cost for doing this? Is it the consumer? Is it the retailer? Or is it the grower? I think we know the answer to that one. And then I asked what the barriers to improving landscape diversity were, and probably I probably don't need to tell you this, but of course it's the high cost of land and the limited space to be able to do it. One of the growers literally had strawberry crops back to back to back with just a hedgerow in between. So there was very little space to be able to manipulate habitats, although they did have some ideas. And then the administrative burdens and time in actually managing this. So a lot of the farms dominated by tabletop strawberry, and of course monocultivation and production methods don't always allow for growers to put some of these things in. And there is this trade-off between crop productivity and a space for nature. Some growers talked about weather messing up their ambitions for changing landscape. So it can be, you can plan to do something, but it gets disrupted by weather patterns. And one grower suggested it would be really good to hire a dedicated member of staff who just took over all of that side of the farming practice, where they managed all the habitats and looked at how to mix them in with crops. And still, even though we've done a little bit of work in the Bespoke project, there's still a lack of, or there's still a feeling that there's a lack of guidance and tailored advice for specific farm conditions. And then also the growers talked about the role of consumers and retailers in all of this. So the retailers are demanding a premium product, which is less reliant on chemical pesticides. And there's a general lack of consumer education that what they were saying is the consumers need to understand that if they want a premium product, with fewer insecticides, they need to understand they're going to get ladybirds in their punnets. And then finally, I asked if you could implement one activity tomorrow that you could do to improve the landscape on your farm, what would it be? So replanting and improving hedgerows was a big win because hedgerows, I guess, are already there, and that's an area that you can enhance. Reseeding headlands with resilient flower mixes, biannual hedge cutting and reduced mowing all came up. And then, of course, growers are increasingly now using alyssum in strawberry crops as banker plants to improve predators. But one grower was very keen that we can be a bit more ambitious than alyssum, and we might be able to look at other plants that perform other roles inside crops. And then some of the growers talked about wildflower meadows and how they were an advantage because of public relations. So they look good when people walk through the farm or past the farm. And there are also retailer benefits. So there's some indirect economic benefits to having some of these habitats in place. But when I suggested, would you remove a couple of, especially with a grower who had back-to-back strawberry crops, would you remove some tunnels and give that over to nature and flowering? That was an absolute no. Now, we have learned that that's really important in pear production, that you can't just have hectares of pear because you can't get enough natural enemies in. So maybe it's something we could potentially think about in the future. But first, I think we need to quantify the benefits of doing it. So the key points, so coming into my conclusions, so key points about landscape complexity that the growers raised were water management is a growing concern, habitat connectedness they saw as the most important thing, and the benefits they see as pest control, pollination, and enhanced wildlife for various reasons, improved public relations and retailer engagement, and I was really encouraged that about the intrinsic benefits that they valued in biodiversity. But the challenges, which I guess are not a surprise for most people here, are land cost, limited space and time to be able to do this, the demand for premium product, putting more pressure on growers to have the optimum fruits, labour always comes up, and a need for clearer guidance on how to do this on farms. So this is part of the AgroBioConnect project, which is led by Alicio Malusa, and there are 6 European countries involved in this and the UK. And I hope to be able to give you some scientific data at the next meeting. Thank you.
Thank you so much, Michelle. I think that's some incredibly positive responses from growers. And I think the whole mind shift from 30, 40 years ago, quite marked. Any questions for Michelle? I've got a question, and that's the size of plantations, because one of your PhD students, Laura Reeves, I think it was, did some work on apples, didn't she, with wildflowers, showing the impact of wildflowers on rosy apple aphid reduction quite far into an apple orchard. But
yes,
how many meters did it extend for the benefits? I'm trying to remember what the figure was.
So that was Charlotte Howard.
Sorry, not Laura.
That's all right. Yeah, Laura worked on pear psyllid, but yeah, Charlotte Howard worked on how far do the benefits of a flower margin on the edge of the orchard, how far do they go into the orchard? And she only measured up to 50 meters, and basically she saw a reduction in rosy apple aphid 50 meters into the crop if you had wildflowers versus no wildflowers. So presumably they were providing ladybirds, hoverflies, lacewings. But so one of the things also I forgot to mention on my last talk is even with the SWD parasitoids, yes, they're going to go and lay eggs in SWD, but they need to be able to fuel up somewhere. They need pollen and nectar, and they need that kind of injection of energy. And some work from Sweden has also shown that where predators get more food, they live longer, they have a higher fecundity as well. So they can lay more eggs and they live a bit longer, so you're getting more out of that biological control. And that's true for the biocontrol that you release as well, hence the alyssum in the strawberry crops. So you need to be able to feed these predators to get the best out of them.
So in answer to my question, that's quite...
did I answer your question?
Yes, you did, in part. That's quite good news. If it extends for 50 metres, because one of the concerns for growers will be how small do they make their plantations? So
there's a caveat to that, though, isn't there? Because that was aphids and that was generalist predators who fly a long way. So if we're looking at parasitoids, in general, parasitoids don't go very far because they're tiny little... I don't want to call them feeble because they're doing a really good job, but they tend to go 10 meters or so. So that's why when you're thinking about wildflowers, don't just think on the edge of the crop, think inside the crop as well. How are you going to encourage those insects to hop from where they're feeding on pollen and nectar to where that pest is? Think about the distance they have to travel.
Okay, and in 20 seconds, can you answer this question online? Will this work lead to an optimized blueprint for soft fruit plantations?
That's an interesting question. I'll let you know that next year when we've analysed the data.
Okay, that was from Andrew Russell at IPM. Thank you, Andrew. Okay, thank you, Michelle. Thanks for your time. And that takes us on to our final presentation of the morning and of session one. And we have now a shift to storage. So many of you will know Richard Colgan from the University of Greenwich and Produce Quality Centre, who spent most of his career working on post-harvest storage of fruits and vegetables and also flowers. Growing Kent Medway again funded a project. I should have just said about Growing Kent Medway, the way the R&D worked with Growing Kent Medway is that the industry identified a problem that they wanted to solve, and they matched up with a research partner. And in this case, we had the industry matching up with Richard, trying to find solutions to improving the storage of soft fruit. So Richard, tell us what you've been finding.
Okay, well, this is actually, thank you, Scott. This is to talk about a project which is actually an innovative funded project.
Beg your pardon, sorry. I thought there was GK&M.
No, sorry, it's an innovative, but there have been other ones since then. But this is a project that was instigated by Richard Harnden, sat in the front here, by Berry Gardens. And we called it was actually called Coolberry. And this was really trying to find ways that we can help to manage the cold chain and storage of soft fruit. This was particularly looking at strawberries, but it's also applicable to other soft fruit and other vegetables and high value crops that require cooling the minute they are harvested. So other partners in this project were JD Cooling, and we were a refrigeration company, Scorpion Vision, who are 3D vision and infrared camera specialists, and Hennig International, who are engineering specialists. So why are we doing this project? Well, certainly the issues around managing this cold chain for soft fruit is that every minute matters. Soft fruit have very high respiration rates. So when we talk about high respiration rates, that's actually metabolism. the higher metabolism, the more moisture they lose very quickly after harvest. Once you take them off the vine, the connection to the moisture, the water supply is removed. So they often will then start to lose moisture very quickly. Strawberries are particularly have a high respiration rate compared to other crops that you might be also growing, such as cherries and apples. And that loss of moisture and high respiration rate can then lead to bruising issues, changes in color, and increased risk of decay the longer you leave it outside the cold chain. So one of the first part of the project, we actually carried out an audit of several farms, different farms, looking at how long it took particular consignments of strawberries from the time it left the picker's hands to the time it got to the fruit got down to temperature. So in this temperature, we're taking it down to around 8 degrees. So this is the time it took, and then it's just a snapshot of a few examples of that time. And what you also notice is that sometimes even in the transit from the picker's hand to the actual pack house, that time it sits in the trailer, you can actually start raising temperatures in the trailer. So as you might expect, in a warm day, that trailer sat there with fruit being aggregated in pallets, and we saw an average between 2 to 4 degrees incremental increase in the fruit temperature before it even got to the pack house. So this can be a challenge in some cases, and also depends on where the farm is, where the pack house is, the journey, and the number of pickers you have, and the time to pick that crop, enough pallets to fill a trailer. All these are variables that we have to consider. So this is just an example of one example where we were looking at the crates. This is a typical example of people packing crates of strawberries on pallets to take to the pack house. And one thing you can see here on the picture on your left is that the crates offer very little airflow through the actual pallet itself. They're basically what you find, especially with the punnet, different varying punnet sizes, that you can actually end up with a wall of plastic which are trying to cool when you get to the pack house. And this is quite a challenge. And I'll show you some data later on when we looked at different pallet crate designs and how that influenced the rate of cooling and the time taken, and also the energy really that it's taken to cool that fruit, which is a real issue in terms of the cost savings what you could actually acquire by getting different crates. But then that also influences the amount of fruit that you can take off in one go off a field. So there's constraints there which I fully understand. So part of the project was we had a very specialized cooling rig set up in our facilities. This is quite unique, made by JD Cooling. It's a rig that allows you to measure between 5 to 10 crates of strawberries or other soft fruit at a time. And it also allows you to start to play around with the temperature differential between the temperature of the fruit and the temperature of the cooler to set differences that allows you to change the speed at which cooling happens. So we can really start to influence the rate of cooling and understand that effect of cooling rate on quality. So the first thing we did, looking at one of the first areas of work, we looked at how does the effect of delaying cooling of getting that crop off the field into a cooler affect the quality. So I mentioned the earlier graph about respiration rates. We were able to use some of the equipment we have in the PQC based here to look at the change of respiration rate that fruit underwent every hour they were delayed in cooling. And you can see there's a general sort of increase between 0, 1, and 2 hours and it plateaus off between 3 and 4. But that does influence the quality of the fruit that you then later we investigate as we traverse through the cool chain. So looking at some of the more nutritional, the more components of fruits, so looking at sugars and acids basically. So we did some measures of the individual sugars, so fructose, glucose, and sucrose, and some of the main acids, in this case citric acid. We looked at how the impact of delay in cooling impacted those, and you can see after 3 to 4 hours, we did start to see changes in firstly sucrose, but then that bit broke down to fructose and glucose, and so there were started to see differences in the eating quality, the taste, I suppose, one could ask over time. But more importantly, weight loss. So for every hour we delayed cooling, we could see an incremental increase in the weight loss that that was observed. So maybe from 1% to 2.5%, but also the variability. So as you delayed cooling, the variability across your consignment also increased. So it underpins the importance of getting fruit cold quickly. And that was measured after 1 day after the cooling, and then after 3 days. But what you can see, most of that weight loss actually was incurred in the first day of storage. So that's a very sort of, you know, very quick change in weight loss that you can incur by delaying the cooling, initial cooling. Other parameters, things with red color. So the graph on your left is actually a measure of red color. The higher value means it's more red. The lower the value, the redness declines. So you can see again, there's an incremental decrease in red color, background red color with delayed cooling. And then the rotting, the botrytis mainly, we saw a general rise in the incidence of rotting, but also the variability in that rotting. It's actually after 3 days at 2.5 degrees and then 3 days at shelf life. So quite an extreme shelf life, but we do have to shelf life soft fruit at that temperature. So what happened next? Well, so the next stage of the project was to try and design a field-based system that could go into a strawberry field and do this sort of cooling straight after being picked. This is quite a challenge. And it's called Coolberry Unit, and it's just produced by JD Cooling. It all happened over COVID, so there was lots of challenges around getting the right bits of the equipment to it and getting it designed. But what you can see here is there's a chamber on the back end of the trailer. The front end has a diesel generator, and the middle bit is the actual cooling system and the control panel. A lot of technology to sit on a trailer in the field, but it was effective. We managed to load a pallet of fruit onto this trailer through a gap in the trailer using a forklift, and then push it into rollers that allow you to push it into the chamber. And what's important about this is that this actual chamber has 4 zones in the chamber that have sets of fans at each zone, and that allowed you to independently cool each zone separately. And that was only achievable by the fact that we had temperature probes sat on the distal end of the pallet, far away from the fan, that could measure the temperature of the air temperature coming off the zones independently. And that fed back to the control mechanism to then push more air into bits that were warmer, which is invariably the central region of the pallet. So this is an experiment we did, and my colleague Karen Thurston was very integral in collecting the data from this project. We had these sort of different zones, and we had a pallet that was, as most pallets are sort of stacked both sort of perpendicularly. So there's different sort of challenges around getting airflow through the crop. But we had these different zones. We measured that sort of different zones in terms of cooling rate. And what we found is that this is an example on the left of the different temperature probes that we put into the pallet across the different zones. We also measured the air temperature coming off the end of it. And then what we looked at was how quickly the effect of cooling rate. So we have a very tight differential between the cooler and the fruit temperature, so 0.5 degree. What you find is that by slowing down that differential, you get a more variable cooling rate across the pallet. So the blue is the back end of the pallet, the distal end. The orange line is a stack that was right in front of the fan. But as you increase that differential temperature to 2 degrees, then you started to tighten that variability across the pallet. Now, there's probably a sweet spot that we need to sort of find out where that differential is. I mean, if you have a too big a differential or no differential, you can just blast chill the fruit, which is great, but at the same time, overcooling can also affect quality by driving off moisture. So understanding that sort of sweet spot in differential is really important. And so we did some trials sending at a time, so just before the Linton pack house. No, it's actually with the Linton pack house was designed up and running. But we sent consignments from, in this case, we have to thank the Clock House for actually hosting the Coolberry unit. And we sent from the Clock House farm to the pack house. And so the Coolberry unit was the graph on the left. A standard trailer was on the right. And what you can see is that the blue bars represent the temperature at the picking, and the orange bars represent the temperature when it arrives at the pack house. So you can see with the Coolberry unit, fruits arriving, it's about a half-hour journey to get to the pack house, or even less. And in that time, we managed to cool the fruit to around 7 or 8 degrees. And that had the advantages of actually allowing that fruit to go straight onto the packing line when there are gaps in the supply. Whereas the standard trailer, you could see that generally there's a variability across the consignments due to various days we did it on, but there was obviously no cooling. So the fruit had to be cooled, blast chilled when it got there. So it's just using some of the commercial pack out information that they carried out. So in some instances, and it wasn't totally black and white here, but certainly the regular transit to the pack house, there were some issues around shelf life quality at the end of the day, end of 6 days, whereas the coolberry unit, the quality had maintained for longer. So the next stage of that was the last year of the project. We actually upped the ante and we increased it, the trailer size. Well, you kept the trailer, slightly bigger trailer, but also we had 3 pallet capacity. And also one of the issues around it was trying to overcome some manual handling issues that you have on farms where often you're picking up trays and you're putting them down, putting them up again, picking them down, and a lot of people were handling trays more than once. So we had at the back of this trailer, and I'll show you the next slide, we had a tailgate system that you could aggregate the trays on the back of the pallet, on the back of the trailer. You see on the right-hand side, and that allowed you to, when you moved the tailgate up, it would then catch a hook, and that would automatically drag in the pallet to the first stage of the chamber. Would then start cooling, and then once you aggregate in the second pallet, you open up the back of the trailer again, and it would it would drag in the second second pallet to this next stage of the cooling zone, and it would take 3 pallets. So just like lastly, got a few minutes, just for a minute or so, just that what we found was looking at trying to understand that cooling efficiency with trays. So we had temperature probes across different zones and trays, air movement, anemometers, as well, measuring airflow. And what you found was that we could find, you drew a line basically across, a very crude line across the cooling rate here. But what you found was we did that, you could find quite a big difference. It's about a 2.5-fold difference in the type of tray that we use. So the trays that you might understand have cut down the sides, had much better airflow across the crop, and there's a 2.5-fold difference between the best and the worst trays. And that's all ultimately energy that you're having to put into that cooling that you could actually save. So ultimately, larger trays with big cutouts do have, you probably get less trays per pallet, which means there's a conflict there with how much fruit you get off the actual field in one go. But there's also a benefit of having, like in the larger trays, that you do get better quality at the end of it. So just examples. And then also one of the examples here is looking at sort of punnet design too. So we're looking at some more novel cardboard punnets on the left-hand side. And the challenge there was the punnets had to be slightly bigger, more sort of had like conical shape to them. And because they had to have a bigger flange on top, so you can actually put the the plastic seal on top of them. And that actually influenced the amount of air flow through the actual gap. So they did actually have poor airflow, the fact that they were slightly different design. And the last stage, just one slide, is the last bit of the project was we're looking at infrared systems. So often we're sticking sort of probes into strawberries, measure temperature. That clearly doesn't fit well with people finding holes in their strawberries. So we had an infrared camera system that we looked at that could actually chart the change in temperature of a whole punnet. And the idea was that would actually go into the unit and start to index various trays, and you could start to do that automatically. That was one stage beyond the project. But actually what we found was that we had to go through a number of cameras to find one that would be less affected by the airspeed, the wind of that of the cooling chamber, because these cameras are quite small cameras. And we had to do quite a lot of work but Scorpion Vision did a lot of work trying to manipulate the back end of the data coming out of the camera. I mean, it's actually a lot of work to actually make it resemble the temperature of the fruit. So we had quite a lot of reprogramming of the camera data to actually show that we can actually track. And in the end, we did manage to do that quite successfully, track temperature of a whole punnet based on infrared system compared to just putting probes in. Thank you.
Thank you, Richard. Any questions for Richard before we break for lunch, or any comments? I suppose my question would be, what happens next? Or is that a question for Richard? Would you like to comment on that? Have you got the microphone for Richard? If he's no, he's
sadly JD Cooling is no longer a functioning company, so it's rather dependent on finding a commercial partner.
But a very positive result nonetheless from the
very
from the
positive
research.
result.
Yeah,
and I'd like to see it, and I've had a discussion with Richard about trying to move it forward, but we're stuck at the moment.
Yeah, okay, good. Thank you very much. We're going to stop there because it is lunch.